Okay, yesterday we left off talking about DNA. We talked about the structure of DNA, how it's made up of a phosphate and a sugar and a nucleotide or a new nitrogen base. We talked about how that nitrogen base has to arrange itself in a specific way. We have 4 nitrogen bases: adenosine, thymine, cytosine, and guanine, and they have to pair. A always goes with T, C always goes with G. Well, now we need to talk about DNA replication. Now, remember, we have to replicate our DNA in the S phase of mitosis or meiosis. Oh my goodness. Meiosis 1. All right. Unicellular organisms just continually replicate their DNA because they replicate asexually. But either way, we have to take this DNA and we have to copy it before we can create a new cell. So we have a 4-step process that we have to follow. So remember that DNA is a double helix. Okay, it doesn't meet at the end, sorry. Remember, DNA is a double helix, and it has your nucleotides on either side, and then they're bonded together with the hydrogen bonds, remember? Okay, so this is our double helix. This is our original state. This is what it looks like. Now, the first thing that we need to do is, if everything is bonded together and everything is all twisted up, we can't do much with it. All right, so step number 1, I love steps, is unwind the DNA. Because we can't do much if it's all twisted up. So that's where we're going to end up with a little ladder like this. And I'm going to go ahead and write in some nucleotides just randomly here. And if there's an A on this side, there must be a T on this side. If there's a T on that side, there has to be an A on this side, because we have to make sure that they match up with their partner. And then they're still bonded together with our hydrogen bonds. So that's step number 1. We just unwound our DNA. Now, we can't make copies of it if it's stuck together, if both sides are stuck together. All right? So step number 2 is to unzip the DNA. Now, we unzip the DNA using an enzyme called helicase. This is an enzyme. What do we remember about enzymes? Enzymes, remember, are proteins that speed up chemical reactions. And breaking of bonds, which is what we have to do here. Remember, we have these hydrogen bonds over here. Breaking of bonds is a chemical reaction. So helicase, remember, anytime we see ASE, we know it's an enzyme. And just in case you don't remember, enzymes are proteins, NS, that speed up a chemical reaction, RXN, that's shorthand for reaction. All right, so we have this enzyme helicase, and they're going to basically break up, so it's going to break the hydrogen bonds between the nitrogen bases. Now, Because I'm going to add a whole bunch of stuff here, I am going to shift this over, but I'm also going to kind of lay the DNA on its side. So let me get my green back. So this is the DNA right here. And we have broken it up now. So it was like this. And now we broke it up into two very distinct sides. Okay, remember from yesterday we talked about 5' and 3'. Remember that is where the phosphate attaches to on the sugar, and that these strands are complementary, which means they're not exactly the same. They run opposite to each other. Okay, remember we talked about yesterday that the DNA itself, the sugar, has a 5, that it has 5 carbons. 1 carbon is got to attach, either the 3 or the 5 has to attach to the phosphate. And on one side, the 5 attaches to the phosphate and it goes in that direction. And then on the other side, on the right-hand side, or whatever, it goes in the 3' direction. And this will make sense why that matters in just a minute. So where the helicase breaks it up, this right here we call a replication fork. And I'm just going to take this. This is going to be the enzyme helicase, because helicase is going to break up or unzip the DNA. All right. And there's a million different kinds of proteins. I'm only asking in DNA replication that you know 3 of them. Okay. And helicase is one of them. Now, step number 3 has 2 parts. All right. So the first part, step number 3, the first part is this strand is called the leading strand. All right, so let me just put these, I'm just going to put these nucleotides here because we're going to need them later. Oh my goodness, I'm copying the exact same thing. I should be putting their bases. All right, so also on a test, on the EOC, they may ask you what is the complement to something. So what I'm drawing right here is the complementary strand over here. So we'll see how this works in just a minute. All right, so this is the leading strand. The leading strand can add everything in because we're making a new strand right here. This is the new strand right here. We can build it in the 5' to 3' direction straight across. And we do this with an enzyme called DNA polymerase. All right, so we have, and the DNA polymerase will attach, and this is DNA polymerase. It is going to add nucleotides in the 5' to 3' direction. All right. So this is no problem. So the DNA polymerase is just going to go from 5' to 3' and add the complement on here. So that's an A, it goes with T. That's a G, it goes with C. That's a C, it goes with G. That's a C, goes with G. That's an A, goes with T. That's a C, or I'm sorry, that's a G, goes with C. C goes with G. A goes with T. There we go. So that's easy. That's sort of step 3A. That's the leading strand. 3A is the leading strand. Now, the problem is when we go to make the lagging strand, this one's going to be called the lagging strand. So 3B is the lagging strand. The problem is that the complementary DNA over here is the 5'. So the 5' of the new one that we're making is over here. All right, so that's where it gets a little bit tricky. All right, so DNA polymerase can only work from the 5' to 3' direction. Okay, but what it wants to do is it wants to do like the leading strand does and just go straight through, but it cannot do that. It can only work in fragments. So what will happen is we have to put down a primer, and I'm not going to get into how that's done, but a primer is just a little piece that says DNA polymerase start here. And then the DNA polymerase will attach. This is still DNA polymerase, even though I made it in another color. The DNA polymerase will attach, and it will have to work backwards. And then we put on a primer, and then it has to work backwards. All right, so it has to go from 5' to 3'. All right, so now what we have is a whole bunch of little fragments, and we call these Okazaki fragments. I don't really care that you know that. I just think it's a funny name. Okay, so the lagging strand, I'm gonna write out what I just sort of drew, is made discontinuously. It's made discontinuously. The primer is added so that the DNA polymerase can attach. Okay, so DNA polymerase can attach. All right, and add the nucleotides and do its thing. But it's going to end up, because it's discontinuous, that means we end up with fragments. And those fragments are Okazaki fragments. So the last step is we have to put these fragments back together. And that's where the third enzyme that you have to know comes into play. This guy, I'm just going to draw him here. This guy is step 4. His name is ligase. And basically, he glues the fragments back together. All right, so in the end, we took one piece of DNA and we were able to make a whole separate copy. All right, we were able to make an entire copy. So on the EOC, one of the things that they're going to ask you is they're going to say, you don't have to write this, but I just want to give you an example of what they might do. So I'm moving this just a little bit. All right, so if you have a piece of DNA that says A, T, C, G, C, G, G, all right, this is DNA, all right, and you need to undergo DNA replication. When you copy this, you're actually not copying it exactly. You're going to put its complementary base pairs. Because using the rule, A always goes with T, C always goes with G. So on the EOC, I can probably guarantee you they're either going to ask you this with, let's say, new DNA. This is the new and this is the parent. So if it says you have a parent DNA that's A, T, C, G, C, G, G, what is the new DNA or the complementary strand? Then it's just going to match with Chargaff's rules. Now, we're going to see this is even going to be more important when we go to make a protein. But for now, all we've done is copy DNA. So I'm going to show you a quick video that I think does a better job of sort of summarizing it in a short way of DNA.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer.
I don't care that you know that yet. I care that you know that a primer is created because that's sort of what DNA polymerase is going to stick to. But we haven't really talked about RNA yet. We're not going to talk about that until tomorrow. So I don't want you to worry about that part right now.
This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers
You don't have to know that.
from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right. So the other thing that I did for you here is I also added what would be like a book sort of picture. So I'm just going to highlight. So this is, number 1 is happening over here. That's unwinding. Number 2 is unzipping with helicase. Number 3A is happening over here. This is our leading strand going in the 5' to 3' direction. 3B is over here. That's the lagging strand. And then the only thing that they don't show you is our step 4, which is a ligase that is going to glue it all back together. Okay? So she did give you in the video one vocabulary word that I didn't, that you may see on the EOC, which is semi-conservative. So if it was conservative, or if it was not semi-conservative, you would take the 2 new ones and put them together. But instead, because it's semi-conservative, you're conserving the original DNA and attaching it with a new copy. Okay? Now, the other thing is that she didn't really talk about is going to be a little bit of a step 5, I guess. So I said that there was just 4 steps. Oh, wait, I don't want to do that. Whatever that did, just undo. All right. A little bit of a step 5, which is, let's see, we're going to call this step 5. This is double checking. So DNA, oops, there's another type of DNA polymerase. Checks for mistakes. All right. Now, as you can imagine, this is happening super, super fast. And so mistakes are made. So before we just say, oh, bye, DNA, we copied you, great, we have to check it. And remember when we talked about how cancers get made, that there are mutations that occur because of a lot of different reasons that result in cells overgrowing? Well, a mutation is, one type of mutation is where DNA polymerase makes a mistake and doesn't catch it. All right? So that's one type of mutation that can lead to cancer and a whole bunch of other things, sickle cell anemia, like a whole bunch of other things. All right? So we have to go back and check our work, right? Now, one of the questions that I always get is, how come when we have to do this Okazaki fragments this particular way, or why do we necessarily need to have a primer? So in eukaryotic organisms, in us, like in people, our DNA is so long, we only copy the DNA in its entirety when we are dividing cells. Okay? So every cell has every piece of DNA that makes you you. And when we copy that cell, we have to copy the entirety of your DNA, which is huge. All right? However, when we want to make a protein, when we need to do something for the cell, make something, we are only going to be copying or using a part of it. So that is the importance of primers. And I think that will make more sense tomorrow when we start to make a protein. But for today, we just basically copy this DNA so we can have a little bit of mitosis and meiosis. All right? So tomorrow we'll get into actually making a protein.